Ćao, jel me vidite?
Da, i vidimo i čujemo.
Ja sebe ne vidim, dobro. Ja smo samo Iors na vezi.
Ajmo da sačekamo još malo.
Vidimo se, čujemo se, čujete i mene.
Da, da.
Da.
Super. Za sada samo Iors na vezi, sačekat ćemo još koji minut, jeste 10:00, ajde da budemo ipak malo tolerantni.
Ja mislim da je akademskih 15 minuta skroz na mestu.
Minut, dva pa možemo da počnemo, samo da vas podsjetim, pošto snimamo i bit će materijal za projekat, moramo na engleskom, tako da sve jedno.
But of course.
A i lakše vam je pretpostavljam.
Da.
Snimanje je krenulo, imamo obavezu da vas obavestim, a kad budemo zvanično krenuli prelazimo na engleski, otvaramo. Evo još 1 minut. Hvala.
Ja samo prvi put koristim sad Zoom na ovom računaru i...
Probaj da podeliš, ja ako ne idem...
Pa već sam tražila da podelim.
Dobro, probaj da li možeš da šeruješ screen, ako ne ja ću ti dati host prava, ali trebalo bi da možeš.
Entire screen, mogu entire screen da šerujem.
Vidi se.
Dobro, znači ne vidite ni, vidite...
Super.
Ovo je li tako?
Odlično.
Jer ja ne vidim što vi vidite iz nepoznatog razloga. Shall we give it 5 more minutes or?
1.
1, okay. You can tell us when to start.
Možemo da uradimo, Ana, jednu sliku. Šta misliš, Ana Đurić, hoćeš da nas organizuješ dok Miljana još nije krenula?
Na onu galeriju?
Pa da odemo na galeriju, da...
Onda moram da prestanem da šerujem, jel da?
Može. Tako. Sad će Ana da nas...
Ajde, please everyone turn on cameras if you have. Last photo.
Za sajt.
Tamara, Luka, Teodora, if you can. If not, I will take now.
Luka, Marko, fix your hair.
Okay, I will take one now. Okay. Okay, Luka is here. One more.
Dobro.
Great, thank you.
Thanks.
Okay, perfect. It's 10:03, so we can officially start.
No, I want the desktop.
We have 2 more people joining in.
Great.
Miljana will start sharing her screen, but I would like to welcome you on behalf of the STEPUPIORS project. Today we have the pleasure of hosting Miljana and Marko, members from IRS, to give us the last project webinar for this 3-year period. We're very happy that IRS, as the coordinating institution, will finalize this project with this great webinar. Miljana and Marko have prepared their slides. We'll keep the presentations around 45 minutes with enough time for discussion. And of course, they will both remain open for your questions after the webinar via email or a call that you can arrange with the both of them. All the material will be available at the STEPUPIORS website. And of course, you can reach out to any member of our team at any point for any further follow-up. So without further ado, I will leave the floor now to our Work Package 3 leader from IRS, Ana Đurić, to present our speakers, and I hope we will all enjoy together for these 2 hours. Thank you.
It is my pleasure to present you our first speaker, Dr. Miljana Tanić. She is a molecular biologist with a PhD in human cancer genetics, gained at the Faculty of Medicine, Universidad Autonoma de Madrid, Spain, if I said correct. She also has a huge international scientific experience. She did her postdoc at Spanish National Cancer Research Centre, Columbia University, Aarhus Hospital, and UCL Cancer Institute. Miljana is investigating the molecular mechanisms of resistance to therapy and the evolution of epigenetic landscape in various cancer types. She's using high-throughput genomics, genetic epidemiology, and bioinformatic methods. And also, she's interested in developing the machine learning-based classifiers as medical support tools in oncology. So it is really my pleasure to give the floor to Miljana to talk today about methods for ssDNA library preparation for study of ultra-short cfDNA fragments. So Miljana, please start whenever you're ready. I think we have some technical issues with Miljana, maybe.
With this title, and it seems kind of unrelated to many things that we and that is very recent discovery that kind of fragments even exist. So it is going to be a very technical talk at moments. In the first part of the presentation, I'll be talking more about the liquid biopsies, just to recap everything and to talk about this novel molecular entity that is being discovered. And the second part is going to be very technical. And this is something that I think will be beneficial to many users who have not worked with NGS or have usually worked with NGS using commercial kits. So going back to the basics of really understanding these libraries, how they are made, because this is what effectively influences what we can get out of it, what type of information that we can acquire. So liquid biopsies, this is a well-known story, but I will just do the quick recap. It is the focus of the STEPUPIORS project for a very good reason, and that is because it recapitulates the entire heterogeneity of the human body and of all of the tumor masses that are present in a patient with cancer, specifically here colorectal cancer that we're working on. And these liquid biopsies, usually in the form of blood, contain all types of cells and other types of analytes that are present. And these are being both secreted or actively released during apoptosis or during necrosis of the tissue. So usually when we think of liquid biopsies, we are obviously referring to the blood plasma. And this is something that we are working with because it's one of the easiest analytes to acquire. It is something that is part of a routine blood work for many patients, and they are used to that. But we should not forget that any kind of essentially liquid tissue in our bodies is a source for liquid biopsies. So these can be cerebrospinal fluids or saliva, pleural effusions, urine, or feces. Essentially, we choose the type of liquid biopsies that makes most sense for the particular cancer type that we are studying. And what is contained with these liquid biopsies? Basically everything. So we have inside DNA, RNA, we have circulating tumor cells, we have extracellular vesicles that are also part of an ongoing project at Aarhus. We have proteins, we have tumor-educated platelets, and obviously we have circulating tumor DNA. That is one fraction of the total cell-free DNA that is inside. As I mentioned, all of these can be used to get different types of information from them, with one difference that the only viable piece of this liquid biopsies are circulating tumor cells that can be used also for in vitro studies. The type of information that we can also get from circulating cell-free DNA is really the same as we can get from any different analytes, such as tissue, fresh frozen, or paraffin. So we can have information on mutations, on chromosomal rearrangements, copy number aberrations, methylation, fragment length. This is something that is also being explored within the context of the project. Gene expression, or even detection of viral sequences inside. However, there is one big issue that is reflecting the abundance of circulating tumor DNA. So there is oftentimes people are confused, especially novices, or clinicians, what is cfDNA, what is ctDNA, and if these two terms are interchangeable. ctDNA composes one small part of all the cell-free DNA, and the abundance, the ratio of ctDNA within the total amount of circulating cell-free DNA in our bodies can range from 1 part in a million to even 20% of the total content. And all of this depends really on the cancer type, on the clearance of those cells, of its proliferation index, of its adjacence to the lymphatic system and vesicles that allows these molecules to exit. So it is really a challenge to detect those. But the potential application of liquid biopsies is enormous, and it can really be applied in any stage of the clinical diagnostic workup. It has huge potential for early detection. It is used nowadays in the clinics already for the diagnosis and prognosis of specific cancer types. It can be used, and there are many ongoing studies that are focused on the detection of minimal residual disease after surgery. It can be used for monitoring of therapy selection, and this is one avenue that we are also researching at Aarhus for therapy monitoring, early detection of resistance and relapse. However, it all sounds great, but as I mentioned, the typical concentration of cfDNA in our bodies is very, very low. So in a healthy person, the typical concentration of cell-free DNA per milliliter of plasma is around 10 nanograms. And this translates to basically only 3,000 molecules of cell-free DNA. And the fraction of ctDNA inside is even smaller. So we are literally trying to find a needle in the haystack. So the methods that we are using, we must make sure they are as sensitive as possible, so we can capture every single one of these molecules, because if we miss them, we get false negative results. So technologies that are used for cell-free DNA detection in the clinics vary from low-throughput technologies such as real-time PCR, digital PCR, to high-throughput technologies such as NGS. And all of these have their advantages and disadvantages and their place in the diagnostic workup. So one method that we'll be focusing today is next generation sequencing and its application, because unlike digital or quantitative PCR, it enables us to query thousands of genes, and in addition, it enables us to get information on copy number rearrangements, tumor mutational burden, and other biomarkers of interest. However, when doing next generation sequencing from cell-free DNA, there are many steps along the way until we get our candidate biomarker or our diagnostic biomarker that can mess with our sample and reduce that little amount of cell-free DNA that we have at the beginning. So basically, sample type is one important choice. We need to make sure that the sample processing follows strict standard operating procedures, that it is handled in the cold chain, that we are using validated protocols for processing of plasma in this case, that we are using the most sensitive DNA extraction procedure because of the procedural DNA loss that can be quite high, going even to the 80% in some cases, or even more. And also in the analytical step, where we can do either regular or bisulfite library preparations, there is an issue of losing the DNA during the library preparation steps, and also introducing of errors during sequencing and data analysis. So, as I said, I'm going to go literally to the basics. And one of the very important things that we did when we started working with liquid biopsies was to choose the method for DNA extraction. So this was something that was being done over 5 years ago. And we did a thorough comparison of 5 different kits for cell-free DNA extraction, so commercial kits. 3 of those were based on column-based cleanup, affinity enrichment, and 2 on magnetic-based cleanup. So here you can see a graph that is illustrating, so the same pool of plasma was used across different methods and with different input and elution volumes, and it gave vastly different results. So this really means that all of the methods that we are using affect our outcome a lot. Effectively, you can see that here magnetic-based MagMax method and QIAGEN had the best cleanup, best yield and best recovery when quantified by qPCR method that gives us accurate estimates of cell-free DNA molecules inside. However, there was one surprising finding when we applied one of those kits. So we selected to use for our studies applied by system MagMax, since it is a magnetic-based system and it can be more readily automated, unlike the columns that are usually manually processed and restricted to only 12 samples or more. So, first of all, can someone tell me what is the typical size of cell-free DNA? Or I can say when I was starting to work in this field, it was common knowledge that cell-free DNA is 167 base pairs on average long, and this is because it is wrapped around nucleosomes and that keeps it protected from degradation. And these are the fragments that are effectively released in the bloodstream. So it can be either 167 base pairs or an integer multiplied of these, so mononucleosomal fractions effective. And then when we analyzed our cohort of colorectal cell-free DNA samples, this was from a previous project, Trace Epigen, we saw something very unexpected. Do you see this little bump here? So this one here is the typical fraction, 167 base pairs. It is beautiful, it is nice. But in some samples, and I just cherry-picked a couple to represent the finding, we see a fraction that is around 45, 50 base pairs long. It looked real. We repeated these experiments over and over, and we saw that we can find them until many freeze-thaw cycles of that samples when they've suddenly disappeared. But in some cases, this fraction is as high as the main dominant mononucleosomal fraction. Sometimes it is lower, sometimes it is the only one present. But it was detected in over 80% of our cfDNA samples. After validating that we are not crazy, that this is something that we repeatedly see, we obviously searched the literature to see, because I have never ever heard of this kind of fraction. I was working in the field for 5 years, and all of the people I've spoken to have never heard of this fraction. Well, unfortunately for us, or fortunately, depends how you see it, this fraction was indeed described at that time in only 2 or 3 papers. That's it. However, now there are more publications coming out, but this is still something that is vastly unexplored. And so in these studies, they have shown that this 50 base pair fraction is predominantly composed of single-stranded DNA. And this is the reason why it was more sensitive to degradation and freeze-thaw cycles. And this is why it just disappeared with repeated measurements. And this only stresses that the importance of properly allocating your samples for QCs in the beginning and as small volumes as possible. Because if you need to repeat something, you don't want to do it several times to unfreeze. So one of the most important studies by Hudekova et al. Did a very thorough characterization of this molecular fraction. So many studies in cancer are focused on this region. It is known, and we are collaborating with one of the teams that discovered that in cancer, cell-free DNA fragments tend to be shorter than the 150 base pairs, and that this fraction is associated also with cancer stage and prognosis. However, when looking at this fraction, it was discovered that it is mostly consistent of portions of the genome that are associated with G quadruplexes. And these regions are enriched in the regulatory portions of the genome. So this is something that has really high functional relevance for gene expression and should be used. And this discovery, in the case of these people, was made only because they were using magnetic-based cleanup and single-strand DNA library prep. Because I mentioned before that we can use column-based cleanups. In column-based cleanups, you usually have threshold, and all the fragments that are shorter than a certain threshold, usually 100 base pairs, are washed out. So many of the groups that were working on cell-free DNA all over the world were just washing out that cell-free DNA inside. That is the first reason. The second reason is people usually don't check the fragment sizes of cell-free DNA, even when they do that. So this is something that can be easily missed. And the third important thing is that when you're doing cell-free DNA library preps, the traditional methods for sequencing that are used are based on double-stranded DNA ligation. And you cannot capture these fragments with that technique. So you would just lose it. And in this study, they combined magnetic-based cleanup with single-stranded DNA library prep, and they compared it to the column-based cleanup and double-stranded library prep. And as you can see here, the libraries around this is the insert size of the libraries, not the fragments itself, and it is a bit skewed to the right by adding of these adapters. It is present in both cases. However, the ultra short cell-free DNA fraction is present only when using single-stranded DNA library prep. And this is basically the point and why I wanted to give this kind of talk, to bring forward these methods that can be used for the study of this very intriguing and potentially very functionally relevant fraction. So now we come to the technical part. As I'm aware that many of our students are initial year PhD students and early stage careers, some of them may not have had contact with NGS technology. So I just want to recapitulate the entire process, because this is something that is very important for the methods development. So the typical library NGS protocol consists of the library preparation, of the amplification or enrichment of specific regions of interest, and this is an optional step, sequencing and data processing. So the library preparation step is the critical step where we select which molecules, which part of the genome we are going to analyze. So we rarely analyze everything. Every method has a bias towards some region. And this is something to keep in mind when doing bioinformatic analysis afterwards. So this is a typical structure of a library. This here is our fragment of interest. So this is what we are enriching. And every NGS library, at least the ones that are based on Illumina technology but not necessarily PacBio or Nanopore, they have slightly different strategies. But Illumina ones, they have adapters on both sides of the fragment that are containing of the P5 and the P7 sequence, that is the one that is ligating to the flow cell, that enables literally anchoring of the fragment to the flow cell and sequencing. Add read primers, sequences that are always the same in all of the different types of libraries, that allows us to go and read our unknown sequence. In addition to this, these are essential parts of the library structure. There are other things that can be inserted in the library. And these can be indices to help us demultiplex sample, and also unique molecular barcodes. And these have very important functions. So unique molecular barcodes, these are just random nucleotides that are used to identify different molecules of the sample. So when we do a PCR of one fragment, it gets amplified to exponentially, obviously, to an extent that we want. But in the process of PCR amplification, there can be an error that is introduced. So when we are analyzing the sample afterwards, we may not be sure if that error is SNV, if it is a real mutation, or if it is just a PCR error. So molecular barcodes, they allow us to do duplicate removal and to kind of collide all of these fragments to the single origin molecule. So we can know for sure that, for example, this is an artifact. These red spots, this is an artifact, and this is the real SNV. And it can also give us an idea of how many unique molecules we have in our library. And that is a proxy for the library complexity. Multiplexing, I guess everybody knows this, but this is this is our known nucleotides in a specific sequence that allow us to identify different samples when we are pulling all of the samples together for sequencing, and this can be done in silico to separate them. So how do we get those adapters onto our fragment? There are many different, not many, but a limited number of different strategies for adapter tagging. The traditional strategy is to introduce these adapters, P5 and P7, through PCR. And this is usually done for small panels and targeted sequencing, amplicon resequencing essentially. And here we can use a complementary sequence to our region of interest, and on the other, a stubbed adapter that can be reamplified afterwards to the full extent in PCR, in the final PCR cycle. This can also be achieved at random. It doesn't have to be specific. So the PCR can contain random primers for both top and bottom strand, and can amplify I wouldn't say unbiasedly, randomly, definitely randomly our library, but with any PCR, there is always a bias towards CG-rich regions. So what we get in the end is not the ideal representation of what was present in all of the molecules in our initiating sample. Another strategy that is widely used is ligation. And ligation initially, in the early days of NGS, was done through ligation of complementary adapters. And this produced so-called non-directional libraries. So you would know the orientation of your fragment. And later on, this was resolved by using so-called Y-shaped primers, where P5 and P7 adapters share a complementary sequence that allows them to be ligated to the double-stranded DNA here. And afterwards, by introducing P5 and reverse P7 primers to be amplified in the correct order. So we would always have P5 starting at the 5' end and the P7 reverse complement on the 3' end of a fragment. Another great way of introducing adapters to our fragment is in a single step by using TN5 enzyme that is performing so-called segmentation. It is cutting the DNA at the same time ligating the primers at both sides of the fragment. So it can skip the initial part of library prep where you have to cut the DNA into appropriate sizes. However, because it does this cutting, it is not ideally suited for cfDNA because cfDNA is already fragmented, although it can be used. So these were the strategies that were used traditionally and most widely for double-stranded DNA ligation. When it comes to classical ligation, it can be achieved also through various ways. So we can have what is used in Illumina protocols is the TA ligation, where in one step of the library prep, we are adding an A overhang, and adapter that contains only one T overhang is being introduced and ligated to the double-stranded fragment. It can also be done through blunt end ligation, so there is no overhang. This is a mistake here. There should be nothing. So it should be blunt end without any T kind of overhang. And here is also a mistake. I took this from from a website, but there is also sticky end ligation when there is a slightly bigger overhang, like in restriction enzyme cloning experiments where you have complementary parts, and this allows for a more efficient ligation. So ligation is a notoriously inefficient process. So only 20% of all the fragments that are present inside of your sample will be actually ligated by both sides. So you lose 80% of your precious little cfDNA that you have during this ligation moment. And there are other strategies that were being invented and other enzymes. So basic research is really important for everything. So enzymes that were using ligation of single-stranded DNA have been applied for developing new molecular biology methods and protocols. So one of these enzymes is circulating ligase. It is importantly a thermostable ligase that catalyzes usually circularization of single-stranded DNAs. But if we block these ends or remove the phosphate group, there can be no circularization, and it can be used for other purposes. Another enzyme that is used for single-stranded ligation is the splintar ligase, and it's used kind of a trick, a splint molecule that is serving as a backbone for hybridization of the acceptor and the donor and the ligation of these two DNA fragments into a final product. So also covering the basics, when we do sequencing, no matter what the strategy that we use to tag those adapters, the next step is to read the fragment inside. So reading of the fragment can be achieved in different strategies and can be done by single-end sequencing, only from forward sequencing primer 1. It can be done in a paired end, and this is usually the strategy that is employed in most of the commercial technologies for mutation detection. Single end is usually done for counting only. For RNA-seq or for short fragments, and mate pair strategies where the libraries are made in a different way, and these are usually used for detection of structural variants and long-term interactions. And one important thing to remember when you are doing downstream analysis, and if you're going to do your own bioinformatics to analyze the data, is that the read one that you get of the sequencer is identical to the to the 5' basis of the top strand, so identical to this one. And the read 2 is identical to the bottom strand. So I know this can be confusing when people start analyzing their data. And now we come to different types of library preparation methods. This is just one snapshot of a document that is 20 pages long, and it is illustrating all the different types that people have invented to make libraries using all kinds of strategies with beads, different enzymes, different sequences. Essentially, this is what is driving discoveries in specific context. One thing that is very important for us working in the liquid biopsy field are the methods that are used for ultra low level detection of DNA. So the methods and the sequencing methods that we use have to be very, very sensitive. Ligation, as I mentioned, is usually not one of them. And one of them, but for single-stranded DNA, there are also not as many methods, but new methods are coming out more and more. So one of the first methods that was developed was this protocol described in 2013 that was designed actually for the analysis of ancient DNA. And the incentive here was ancient DNA is present in even more scarce amounts than cfDNA. It is usually degraded. It is oftentimes has single-stranded overhangs, long overhangs, and it was really, really hard to get any kind of useful information using traditional library prep methods on ancient DNA. So these guys invented a method that was using at the time completely different strategy to generate libraries. So for example, they would start off with a typical damaged DNA fragment coming from some, I don't know, bone that was lying there for millions of years, that contained both nicks inside, that contained blockers or uracils, all types of damage, long stretches of single-stranded DNA. So the first step in their protocol is to remove the 5' and 3' phosphate groups to prevent circularization. This is something that I was mentioning, and to allow further adapter ligation. And then the step to do heat denaturation, so all of the molecules can be separated into single-stranded DNA. I'm going to move this here. So the next step is adding phosphorylated adapter that contains 3' biotin, and using this Circ ligase enzyme to ligate it directly to the single-stranded DNA molecule. So this was really an innovation here. And afterwards, the procedure becomes more familiar, and they're using immobilization on streptavidin beads, and they're using a phosphorylated, they're using an adapter to extend the fragment, and then another step with the classical T4 DNA polymerase, removing the overhangs and ligation, blunt end ligation of the second adapter that contains terminally blocked and with a D-deoxynucleotide. After that comes washing and separation from streptavidin beads, and final amplification with the desired number of PCR steps. So in the end, they get a full library that contains both P5 and P7 adapters. You'll notice here that this part P5, P7 adapter and the read1 or read2 sequencing primers don't need to be one next to each other. In between, there can be all types of sequences like barcodes or here just adapter sequencing, essentially prolonging that chain. So that can be, once you have the final library, it can be directly sequenced or it can be used for target enrichment. Sorry, next. And this was a method that was developed in 2013 and in 2020, there was an update of this method, enabling both automation and improvements of some steps, so making it even more sensitive. So one modification is here you still use heat denaturation to render all of the fragments single-stranded, remove the 5' and 3' phosphate groups. But instead, here they are using a splinter oligo with random 8 nucleotides to enable the ligation of the adapter that has biotin on its 3' end, and also to afterwards being immobilized on streptavidin beads. The splinter is being washed out, and there is an extension of the full sequence using standard Klanov DNA1 polymerase fragment. And one extra step that they are using from here is to determine the precise number of PCR cycles, because you don't want to over-amplify your library. I'm going to show later some example how that looks like. And afterwards, washing and again, blunt end ligation of the second primer and final PCR amplification. One thing that can be done is to select also the proper size for sequencing that is most precisely done with gel excision on a page gel, or it can be done on agarose gel as well. Or clean up with spray beads, which is less precise. Yep. So these were like homemade methods that were initially developed, and afterwards, many companies, they came out with their own strategies for single-stranded DNA library prep. So I'm just going to mention a few of them, but if you Google single-stranded DNA library prep, there are at least 10 different providers, some of which are completely unknown with their own protocol, but they are usually based on the same type of strategies. So Takara is a very well-known company that produces high-quality reagents and has had some very good kits for ultra-low DNA library prep, and they were adapted and changed also for single-stranded library steps. So again, the pre-treatment denaturation is always the first step in the case of intact DNA. Afterwards, they are doing adapter ligation to the single-stranded DNA. I think they're using their own enzyme. Methods for details of a commercial methods are not always fully disclosed. So if you want full details of a method, you can usually go to the patent if it is patented. That is also one thing. Sometimes companies do not want to patent something because then you can reproduce it at home, essentially, not at home, obviously, in the lab. But it is a protocol that ligates single-stranded DNA and afterwards generates a complementary strand, and again does a second adapter ligation to the 5' end, and after that the PCR to amplify the entire structure of the library. This is another not so known. I just cherry-picked some of the commercial methods that I found that are some very well represented and some less. This is the producer that I have heard for the first time, but they are using also a very interesting strategy for single-stranded DNA that can be applied both to cell-free DNA, shear DNA, or bisulfite-treated DNA. So what they are doing is they're doing extension by adding poly-C tail and adapter that is complementary to the sequence to extend it, and then they are doing second adapter ligation. It's not drawn here, but it has to be, I think, on the double-stranded DNA to produce the full-length library. This is I don't see the manufacturer here. Sorry, I forgot to put this one. But you can see that the strategy is very similar. So most of these new products are using the same strategy. And what is important is that these protocols are quite quick. They usually take 1 day to prepare the entire library. This is another new producer that has the same, practically the same strategy for preparation of single-stranded DNA libraries. One of the first kits that came up almost 10 years ago was from Swift Biosciences, and it was acquired by IDT. So this is the current name. Is using a different strategy for their library prep. So they obviously start again with single-stranded DNA fragments that were made either by bisulfite conversion or by heat denaturation. And they have a proprietary technology, an enzyme that they call adaptase. So it is hidden behind a patent that creates an extension, we don't know what kind of nucleotides are being added, but it's being used, the so-called adaptase, for ligation of the second stubby adapter, or the first stubby adapter. It is then extended through usual means clonal fragment, and another stubby adapter is then being ligated and using PCR extended to the full index library. So we tested this library. The cost is reasonable. Usually these libraries are around $100, euros per sample. In Serbia, probably more. And I just like to show you something that happens when you have too many PCR cycles or too little of your DNA template. So this is the same cell-free DNA that was amplified using this library. And here we use 30 nanogram input, here 10, 3 nanogram, and 1 nanogram. So this is 3,000 molecules. This is 300 molecules that we have inside. And if you have low input, low template, you usually see something like this, double of your actual size. And this is called daisy chaining or bubbles. So if you see this, don't be afraid, because these libraries are perfectly sequenceable. They are just like two templates that start amplifying each other, but you can sequence them in a regular way and get the same results as if you would see this type of library inside. Interestingly, I did not notice this in the beginning when I analyzed my libraries, but you see here this little bump, maybe? So this likely corresponds to our 50 base pair fraction. These are the markers over there. So all of these methods can be applied as well for methylation sequencing. And this is interesting because methylation is one of the biomarkers that is very the subject of a lot of research for liquid biopsies, since it's promising biomarker for early detection and monitoring and other purposes. What is the common trait here is that after bisulfite conversion, DNA, cfDNA, is effectively made into single-stranded DNA. And then a number of methods were designed specifically for preparation of these types of libraries. So one of the first, and these libraries are also can be used on single-stranded DNA. So everything that works on bisulfite converted works on single-stranded as well. Here is the representation of a traditional kind of workflow for whole genome bisulfite sequencing, where fragmentation is done initially, adapter tagging is done through ligation, followed by bisulfite treatment, where you destroy half or more of your libraries, and then you amplify the rest, what you had. PBET, that is short for post-bisulfite adapter tagging, changes this by first performing bisulfite treatment that effectively cuts the DNA, and then adapter tagging. So that is one strategy that can be achieved. And this is achieved through this method. So random primers, again, labeled with biotin, so there can be no extension at this end, are being used in 2 rounds to amplify randomly the entire sequence. And it is a method that is suitable for ultra low input material. So we're talking picograms. Another interesting method that came out was using, again, a completely different strategy for library preparation. And this was called capture amplification by tailing and switching. So again, you would start with short single-stranded RNA or DNA, doesn't matter how it is produced. And then you would use poly-A tailing and an adapter that has a poly-T tail and a 3' stubby end. However, the enzyme that is used here for the extension is not, it's not polymerase, it's not kind of fragment, it is MMLV, it's a reverse transcriptase. Since it was noticed that specific types, most of the types of reverse transcriptase have the ability to, after the extension, to introduce this 3' CCC overhang that is usually 3 to 5 nucleotides long. And when provided with a oligo that contains a complementary sequence here, they perform a template switch. So they jump from this one to this one, and they start extending in the other direction all the way here. So you have one-step library preparation method here when you introduce both enzymes. And this is very, very efficient process, since this is the mechanism that is used in retroviruses. So to amplify retroviruses to reverse transcription, it can also use DNA as a substrate equally well. And I'd like to show you here how this works. And this was done on a plasma sample, same plasma sample at different concentrations using the same number of PCR cycles. And here you can see a beautiful library made out of 30 nanograms of DNA. And this is with 10 nanograms. This one was made with 1 nanogram. I have one, it's the wrong one, but there's also libraries that can work with 300 picograms. So you can see here we're going down to the picograms, and it is linear amplification. It works like a charm, and it is an in-house method that can be made by $10 per sample. You just buy the enzymes that are used for the reaction. All of these methods can be, once you make the library, they can be combined with hybridization capture, so you can enrich for the regions of interest. If you don't want to do whole genome sequencing or whole genome bisulfite sequencing, it doesn't matter. However, you must make sure that the probes that you are using, if you're using bisulfite conversion, are compatible, that they are for bisulfite converted sequence. And if you're using regular DNA, then the normal capture probes are perfectly fine. Other strategies for hybridization capture for ultra-low inputs, starting from single-stranded DNA, can also be through these bisulfite or regular padlock probes, molecular inversion probes, they're also called. And they're commercialized by Agilent as Haloplex, so that you can order your own sequence. And this can be done on single-stranded DNA after denaturation. Or this was a protocol that was adapted by using any type of these single-stranded library prep methods and biotinylated probes to pull down all of the fragments of interest that can be afterwards sequenced. And finally, we're approaching the end of the talk. So this is maybe too much, but for those of you who will be working with data, I think it is very important to understand, especially if you're going to be working with methylation. After bisulfite conversion, you effectively and PCR amplification, you have first two non-complementary single-stranded. I wouldn't call them DNA molecules afterwards. They are a mix of, they look more like RNA since they have uracils inside of ctDNA. But after amplification, they are converted to cytosines again. So you have two different combinations where you have the original top, the complement to original top, original bottom, and complement to original bottom. So you have 4 possible different sequence strands from one genomic locus that you need to map. And knowing what strategy for library preparation you have used directs how you're going to bioinformatically process these the data that comes out from the sequencer. So you really have to know if your libraries were directional. That means that read 1 is going to be here, that you're only going to have read 1 and read 2 corresponding to the original top and original bottom. Or if you're using any kind of random priming strategy, such as PBAT, you're not going to have original top and original bottom. So you may have like 0% mapping efficiency. If you expected this, you have to use specific settings in your pipeline. So you would have only in case of these random priming original top and original bottom for these types of libraries, or you're going to have non-directional libraries, such as the initial ligation that was done without the Y-shaped adapters, or some other strategies that will give you for all 4 different strands. So you would have to align to all of the combinations. And that I'm not going to go into this. I'm just going to leave this for the talk. If anybody wants to understand more deeply how these look like, they can have a read. And that is all from me. Thank you very much for listening. I know this has been a bit more technical. I think Marko's talk is going to be more interactive, but I think that I hope that I have transmitted the message that methods matter. You need to know the tools of the trade that you are using, because if you're using inappropriate tools, you may miss out on important findings, first off. And if you don't understand how they work, you might misanalyze your data and again miss out on information and discoveries. And that would be a shame for the time that you invested and resources and everything. So thank you for bearing with me, and I'm happy to take any questions if there are any. Thanks.
Thank you, Miljana, for your wonderful and detailed talk. If anybody wants to start with the questions, if not, I have one that is maybe technical.
Yeah.
But as you said, how does the choice of extraction chemistry that you use impact the representation of cfDNA in the libraries?
A lot.
Why it is important?
And so, depending on, this is something that translates to the utility of that cfDNA in the clinics afterwards. So if you're using a method that has huge procedural loss, you won't have good complexity of those molecules, and you may miss out on very rare allele frequencies. And when we did the benchmarking, we could see almost five-fold differences in yield from the same exact plasma sample between methods. So I think very important part of any kind of project is to choose the appropriate method for that. In our hands, this applied biosystems magnetic-based cleanup works the best, because magnetic-based cleanup, it just captures all of the cfDNA molecules inside. There is no the threshold is actually made by the binding buffer there. So there is an option to maybe use more dense binding buffers to capture all of the molecules. So I think there might be even room for improvements in the methodology so that we can enrich for this fraction.
Okay, thank you.
Thank you.
Anybody else?
Can I ask two short questions? Miljana,
Yeah.
I won't bother you a lot. We can talk this. So, of course, all your lectures actually inspire us to search further on for the basic science approaches. I think this you've highlighted very, very well why this is important. And of course, we always like to talk about the general picture of what we want to achieve, but when you go back to the basics, you of course need to take care of both the sampling and the pre-analytics and everything, because it affects the level of conclusion. So this is, you highlighted it perfectly. I was exploring a little bit. I was surprised to see that Oxford Nanopore is actually developing the technology for ultra short, because they were traditionally for the long reads and for the long fragments. So what do you think? Is this new technology? I see they published something in 2025 in April.
Yeah. But
So
what
do
they
you
call
think?
ultra short is aimed for the typical cfDNA size. So this is ultra short for them. It's going from 10 kb size to 100 base pairs. I'm not sure. I mean, in theory, they can do 50 base pairs. They can do anything. If it
It
goes
says
to
they go
nanopore,
under 40 base pairs, but I don't know, because we've explored it a little bit, and I mean the publication is there, so everybody can review it. But
yeah, I was looking at that as well, most importantly because it gives you simultaneous information on genetics and epigenetics,
Yeah.
and that is a huge value from nanopore. And it is also a direct method where you don't have any conversion of the original molecule. Again, you have the representation unbiased of what's inside. I think it's a matter of cost effectiveness for the moment, because this method has high cost per sample. And I don't know, I don't have any data on how efficient it is for cell-free DNA in comparison to other methods. So
Yeah, me
that would be
neither.
my kind of
Yeah. Well, let's
worry.
I'll put the publication in the chat so people can review it. I think for
Thanks.
the young people, it might be interesting just to compare. You've really presented a lot of technologies, and it's always a matter of choice depending on where you are and what you want to achieve. And the other thing I think which correlates well with what you said is the impact of this library prep on what you are going to detect. So this is, I think, the issue. What about the effect on the diversity of things that you are going to see on early detection, on everything? What do you think? Is this crucial as well, or?
It is. So I didn't mention that much. I didn't went into the biology, but for example, these ultra short cell-free DNA fraction seems to be enriched in healthy individuals. And but it's controversial results, and it can have use. It's also enriched for regulatory regions. So there are some publications that came out trying to use it for early detection and for diagnosis as well. The only issue is that in healthy individuals, when you extract cfDNA, there's so little of it to start with. So if this fraction is lower than the dominant ones, not sure. I think we have to improve the methods even further. Take maybe more samples from healthy people. We can afford to take more than 10 mLs of blood. And also see if this is maybe available in other types of liquid biopsies. Do we see that? In what context? We know very little of it. So we know which portions of the genome it represents to, that it is often enriched.
I think we have technical problems.
Miljana is frozen, at least for me. Yes,
Okay,
sorry.
you were frozen
You're
for
back.
me. So.
Now we're here.
Yeah, I just wanted to say that I think for the early detection, this would be interesting to explore. It's now it's always been a hot topic, but in these recent years, everybody is trying to find methods for early detection of cancer. So I think if somebody here who is listening and wants to collaborate with Miljana on this, please feel free to do it, because it will be a treasure trove, I think, in the coming years. But technology will be the main issue here. So Miljana, you're the right person to work on it. Thank you for everything.
Thank you. And I can stop. Did I stop sharing? Yes, I did. Okay, good.
Okay, thank you, Miljana. Now I am happy to present our next speaker, our dear colleague Marko Radulović. Marko is actually a pharmacist, holds a PhD from the Faculty of Pharmacy, University of Belgrade. He also, as Miljana, has a huge international scientific experience. He did his postdoc at Max Planck Institute in Göttingen, Germany, University College London, and Imperial College London. Recently, Marko is a research professor at the Institute for Oncology and Radiology of Serbia here in Belgrade, and his research focuses on the investigation of molecular and morphological prognostic and predictive markers in cancer. Actually, Marko is a pioneer in using AI and machine learning models at our institute. So I'm really happy that he will talk and share his experience about using artificial intelligence in precision medicine and what are actually the advantages of deep learning approach. So Marko, please, the floor is yours. Please unmute yourself, Marko. We cannot hear you.
Unmute. Okay. Okay. So I'll tell you about the use of artificial intelligence in precision medicine. Artificial intelligence actually covers both machine learning and simpler versions of machine learning, but also deep learning, which is probably
Sorry to
real
stop
meaning.
you. We have an issue with, I see at least an overview slide here. I don't know how the others see it. Can you put the full screen of the first slide, please?
Yeah.
Now, perfect. Thank you. Thanks.
Yeah, so I will not go into exhaustive theoretical coverage. It's an enormous area of research. I just want to give you the most basic facts that will allow you to even start a very powerful machine learning platform today, if you want, with your own images and your own features, which is quite amazing, because people struggle with deep learning and use of artificial intelligence for a single reason, that there's no software with graphical user design interface. So the work is mainly in raw code. So why is that the case? I don't know. It's would be easy to actually produce a graphical user interface for even for the most complicated neural networks, but somehow it's now the privilege of engineers and coders to play with that, and then you have obviously a very strong gap that you don't know how to code, and then you call the engineers, but they actually only have experience in recognition of car register plates and faces, and when you show them MRIs and histopathology, they don't know what to do with it. So then you're lost, basically. So Renaissance personality, which integrates both, is not only you know something to you know, a theoretical goal, but it's actually a necessity probably in this case. So first I want to show you the green slides are about basic facts. So what is risk stratification? So people actually get lost in this, you know, terminology and think that it's something really complicated. But actually, risk stratification is just ordering numbers, either in their ascending or descending order. And you can use Excel. You don't need any statistics to do risk stratification. Because, well, simply, although it is quite, sounds very complicated to use deep networks, or you use complex machine learning platforms which produce tens of thousands of models, but in the end, from each of these platforms, you get a probability score. That's a continuous number. You get only one continuous number per patient. And if you order these continuous numbers, oops, uh-oh. Now it's not working in the, yeah, if you order these continuous numbers, so for example, if you just put randomly order your patients, the positive patients, let's say metastasis positive or whatever positive outcome, would be randomly distributed, as you see here. But if you order them by whichever score, continuous score that your AI or even molecular or whichever actually platform predicted, you should see some kind of stratification. So this is stratification. So you see more positive cases on one side than on the other. And statistics is here only to formally determine whether this is significant or not. So that's is that easy, actually.
Sorry, Marko, can you put a full screen?
Yeah. I'll go back to full screen now, but yeah, I couldn't activate the Excel sheet without going off full screen. So precision medicine applies to many different things. It can apply to prevention, where you, as I say, measure BRCA mutation and then apply some preventive measures to prolong life of these people. Or during disease, you can monitor biomarkers, circulating DNA, and things like that, a bit like what Miljana was talking about. But I will focus mostly on prognosis and prediction. So prognosis in the most common terminology means guessing the disease outcome at the stage of diagnosis, when disease outcome is actually unknown. And prediction is therapy outcome before its application. Although these two terms are mostly regarded as synonyms, so in this area of research, they have different meanings. So, and another, very major rationale that I want to demystify is why we need precision medicine. What's it all about? So the precision medicine is simply a trick to get more out of classical therapy, because cytotoxic therapy has limited benefit in cancer. So improved survival needs novel drugs, and this takes many years to develop and even more years to register. So precision medicine aims to optimize the use of existing drugs to prolong life of patients. So that is quite simple, well, which is not very obvious. You know, if you read the literature, you could, remain mystified. What does that mean? So you basically predict which are high risk and which are low risk patients, as I saw you by these scores that your model quantifies. So because risk stratification, in this case, where you want to guess the outcome of a disease or of therapy, you're essentially guessing the future. Yeah, and that's why molecular methods, which are very preferred today in every sense, and work wonderfully, and so, but in this area they don't. They have very poor performance. So the genetic profiling of 200 genes, such as MammaPrint, has an ALK of 0.65, which is quite poor performance. So it seems that molecular analysis works well for determining the current state of the organism. It's not good of guessing the future state of the organism. So that's why you need esoteric methods. Could use tarot cards, tea leaf reading, but yeah, in science, you can more easily publish if you use morphology analysis. So such as radiomics and deep radiomics and deep learning. Although you be still considered a weirdo, but you can sort of make a scientific rationale that why do we want to do such strange things, like look at how the tumor looks like instead of measuring molecules. So the thing is that I've been doing, for example, proteomics for quite a few years. Proteomics is, now measuring thousands of proteins. But the problem is, let's say 5,000 or 6,000 or 7,000, but there are 20,000 proteins, and there are many, up to 1 million of modifications. And then it turns out that you actually measure probably less than 1% of all proteins, protein and their modifications in the cell at only one time point. So if you want to measure real time, it probably would be possible in several hundred years. It's like we're really far away, although we are kind of measuring thousands, and we think that we are, put omics to it, and now we can, do really a lot, but it's very limited methodology. So then morphologists make a point that actually we can again circumvent all the shortcomings of omics or genomics, proteomics, and so on, by saying that morphology reflects the sum of all molecular interactions within the tumor. And then if we measure morphology, we measure all million of different proteins and all who knows how many lipids and carbohydrates and DNA and whatever there is, and however a number of cell types interact. So the final sum is actually the tumor morphology, and the tumor morphology can that be used for reading of the future behavior of the tumor, and most important, actually, tumor behavior is whether it will give metastasis or not, because most tumors are by themselves not very actually a big health problem. It's the metastasis that kill the patients. So that was a very important thing. So of course, you probably know most of the standard precision medicine or prognostic markers such as tumor size, histological grade, the hormone receptor status, and all the genes that are analyzed. But as I said, as I mentioned, this does not work very well in terms of prognosis and prediction. So in terms of morphology, the emerging approaches are morphological, and they use MRI, CTs, and histopathology in three main versions of methodology, which I will explain later. They're kind of similar, but also very different: radiomics, deep radiomics, and deep learning. So, yeah, even in 70s, the morphological analysis of tumors started with only like several factor features and several textural features, and even these actually had better prognostic performance than the molecular prognosticators. So radiomics is actually a very recent methodology, but it's turned obsolete kind of very quickly because of the occurrence of deep learning. Actually in the same year, but deep learning took slower to take off, but radiomics took off very fast. And but radiomics is a very classical method in terms that it uses predefined mathematical formulas. It actually just increases the number of textual features. So if we use 5 features or 10 features previously, now it can use thousands of textual features, but they are predefined. So deep learning is revolutionary in the sense that there are no predefined mathematical formulas. It doesn't calculate so many features. It's up to 1,000 per network. So it actually also calculates textual features, but in a different way. It does it automatically, and then it calculates it taking into account the classification. So it is a much more advanced methodology. So actually deep learning is a part of machine learning, and yeah, machine learning is simple, uses simple little tools like support vector machines, random forest, k-nearest neighbors, gradient boosting machines are new ones, and most famous feature selector lasso. Again, that's all very nice, what I said about both machine and deep learning, but the problem is that it's not made for use by an average scientist, or actually by any scientist which is not who is not a programmer. So this is a very essential fact if you do any kind of machine or deep learning. Yeah, of course, this sort of machine and deep learning is not specific for morphology or for MRI, CTs, or for radiomics. It's not specific for morphology. It can be done on any molecular parameter. So yeah, I don't want to make an impression that it's specific for, you can measure whichever molecules, and you always get the numbers, and then radiomics features also numbers, so the machine learning platform would treat them the same. Also tumor size or measuring whichever tumor marker in blood would be treated the same, because they're only some concentrations, some numbers, and whichever radiomics feature. So you would have a development set which actually divides randomly into training and validation set. The training, it trains on subset of about 70%, and then it validates on the part that it hasn't seen, which is very important because these advanced techniques actually usually achieve 100% accuracy in the training set, and then that is not that is not a problem for them. The problem for them is to generalize to an unseen subset, let's say. And this generalization is even further tested on a test set, which is again a separate additional 30% of the samples, where it could be 20, it could be, it's not very strict. Ideally, for it would be an external cohort to check how your system or model generalizes. But it can be internal. It can be internal. So we recently published a paper with internal test set, which is seen as weakness, because today external test sets are thought to be like the thing. But we made internal test set, which is chronological. So the test set was actually made of newest samples, which simulated the situation that we actually prognosticate metastasis occurrence in the same institution on current patient based on previous patients. Yeah, that makes sense. But it is less, so the performance is not as good as with random actually internal test set, because the chronology means that something changes, like past and more older and more recent patients differ in some sort of protocols. You don't even know how they differ, but they differ. But it gets inferior prognostic performance, but it's more realistic. And now I'll show you the famous actually scientist-friendly platform, which was made in 2020. China, Shanghai, by Siemens was involved also. There are several of these platforms, not many, but I tested them all, and only this one delivers sufficient amount of detail for each model that you can use it for ensembling, and then you can actually evaluate it reliably, because it gives very, very detailed, produces a folder of several gigabytes in size with actually all detail what it did. So that is actually essential. So on this link, you can just actually download it, and you can use it even without installation, so it pops up. So it's not even scientist friendly, it's even iOS friendly, and you can use it on iOS computers. So yeah, what it does. So there are several steps in machine learning. First, you do the data balancing, because if your outcome is 10% of total cases, you need to balance these in some way. So I will not go into detail. So normalization is a very obvious need, because one feature would have values about 10 to minus 20 range, and another feature would have a range of 10 to plus 20 range, and then you want to normalize it usually between 0 and 1. But there are several normalization methods, and they do affect actually to some extent the final result. So then you have pre-processing, which you will see in the next window, feature selection, classifiers, and then this platform uses several of methods in each step, and then combinatorially comes to up to 9,600 models. It also, which is quite amazing, gives you a possibility to limit the number of features for each model. So I limit them at 8 usually, because the more features, the less generalized is, as the more artificial is, the less stable the model is, and usually has worse generalization. So 8 is a number which is quite maximal, but although people use 30 also. So this is what you get when you open this Feature Analysis Explorer. Phi is gives you a very simple graphical user interface where you load the training set, you load the test set, and then you choose for each step. So it's a pre-processing. You can use a PCA, you use a PCC. I check them all actually. This one, PCC, actually removes features which are correlated over the coefficient that you define. Then you can choose feature selectors, classifiers, and the learning actually occurs at the feature selector and classifier stage mostly. And what you need, so now we come to a practical part. When you need, the first column should be patient ID, the second column should be outcome, and the rest of the columns, no matter how many you can have 20, you can have 2,000, are actually features. So they're just features. You have one number for one patient. So patients are here. So you see there's a endless number of features here, and then I would check. I would check basically everything. Let's just speed it up somewhat. So it tells you, which is very nice. If you check this, we'd have 3,000 models, but depending on how many tests or possibilities you check, you'll get a different number of models in the pipeline. So I go for 1,680, which I found is quite sufficient. And then you just print, run and save. We actually loaded the test and the train and test set, and it's that simple. So you can see how it would take probably 2 hours for on a quite average computer for this. This doesn't. It's not proper deep learning, so it doesn't need graphical cards or anything, any fancy hardware. It's and once it's finished, you open the folder that it made in visualization, and now it gets interesting. Only now it gets interesting. So what you get is 1,680 models with all the data, and there's even more data in folders. So you can sort out. So in a training set, you see the AUX are 1.0, and it's all ideal because it's these machine learning tools can achieve 100% in training set. But what we are interested in is test set, because in test set, obviously the results are much lower. So here it's 0.65, but this is one of the cohorts where these are unusually low. So this should go to be much higher usually, but doesn't matter. This is good for demonstration. So actually, by clicking on the top model, you can see its performance in training set, which is good here because the training set should have the highest performance. And then the validation and the test group could have the same or similar performance, but it's not good if, let's say, test set has much higher performance than a training set. That means that we just discard those models. So this is good that it has this visual control. That's quite essential for this platform, and that's why I recommend it, because we won't take all models which have a very good result here in test set, but only those which would make sense. So that is quite important. And yeah, this is still okay because this is consistent. All three curves are on the same side of a graph. Well, in some models, the curves will not be on the same side of a graph. And this is obviously discarded because somehow it's happened that it has a good performance in the test set, but it's actually quite random. It doesn't make much sense. Okay, so that's about it. So this would essentially yeah, enable you to make your own Excel sheets and just do it on your own. Yeah, if you need any help, which is usually the case, you can contact me. So what I did, because the performance of individual models is not so great, so I did assembling, and ensembling sounds quite complicated, but it's actually just integrated the probability scores. Let's say we could get the first 10 or 20 or 15 models that you saw, and just average the scores that they delivered, and that's it. And then we get the ensemble gives us, instead of 0.65, gives us 0.86, which is much better. But it only gives us that with the clinical pathological and radiomics features. So you can load any type of features into this, which is good. Yeah. So you don't have to do radiomics at all. You can just do molecular. Just repeating myself. It's not a radiomics mumbo jumbo. You can do even decent molecular biology, like what nice people do. And this is a deep radiomics, which gets a little more spicy because you use proper convolutional neural networks, and which are red hot now, and they yeah, usually have, this is just an illustration, they have 50 layers or 20 layers, the simpler one have 20, and they have an output layer which makes them completely independent, so they will classify also automatically everything, but deep radiomics actually stops here. It doesn't want network to classify. It just extracts 1,000 features from a deep network. So that makes it a bit like radiomics features are calculated by PyRadiomics, and this somehow adds to that PyRadiomics. And therefore, we still need this machine learning platform that I showed you in the previous slides. This Phi platform will still be useful because we disabled the classification platform of the neural network itself. So here we would have radiomics and network features. So if you take 10 networks, and let's say in radiomics, PyRadiomics calculates 2,000 features, and we have 10 networks which calculate, let's say, 900 features each. So we end up with 11,000 features. So this Phi would work much longer for 11,000 than for 2,000 features, but it still it would deliver better results. Yeah, the more sort of feature providers you have put into this machine learning platform, the better results you get. So I love the names of these deep, or actually not all of them are really deep, but EfficientNet definitely is. DenseNet, ResNet, Exception, Inception, RegNet, Inception, ConvexNet, MobileNet. So you see, of course, the radiomics gives you some kind of feature name, which links you to formula, but here you don't know how it calculated anything. It just gives you from middle layer, this is feature 0, feature 1, feature 2. So it is a black box. So even radiomics is a black box, so you can't avoid that. People want explainability these days, but that is just not possible, I think. Yeah. So we came to deep learning, which is a proper AI and doesn't use any radiomics features. Although you can load, you can still load. Here you can be really creative and just do whatever you want. And you can load this network. This network will calculate its own features, but you can, on top of that, load molecular features, radiomics features, clinical pathological features, and whatever else you want, and it will put it all together and work with everything. That's it's quite amazing, and you can do that all. You can change, you know, with a few lines of code, you can just do whatever you want. You can make a totally new network architecture again with just, I know, like a keyboard and a very average computer. Yeah, so the essential thing here is, which is one of the major takeaway lessons, is to use we use pre-trained neural networks. So that was the case also for networks that were used for deep radiomics, because networks have a very tricky demand of learning on a huge number of. They learn better when you provide them with huge number of images or any kind of data. So these are pre-trained on ImageNet, which has 15 million images. The problem is that these images were for real world, and we actually are working with MRI and CT and immunochemistry images, which are still from real world, but are very unlike real world. And but there's still no one managed to get millions of MRI images and pre-trained on that or histopathology and so we work with networks pre-trained on normal images. And the thing with pre-training is that these you just can use several hundred or even 100 images. The more you have, the better it is, but you don't need a massive amount of images. So with pre-trained network, you only do so-called fine-tuning. So you adapt the network a little bit to your MRI or CT or whichever images, which is nice. Here we don't use any machine learning platform, but we just use the output fully connected layer of the deep network. So another actually crucial information is because the networks are so hungry for information to be trained. Even pre-trained network, like if there is some kind of if there are more images, so that what people do is that they virtually increase the number of images. That's a lazy thing to do. Yes, so you rotate, flip, scale, crop, and you know the same images, and then out of, let's say, 200 images, you create 2,000. And you blur them a little bit here and there, inject noise, and you trick the network into thinking that you actually have 2 or 3 or 5,000 images. Well, this is still not the real thing. So we said, well, we don't want to do that. We don't want to do it properly. And actually, we made a major kind of, almost, it's not breakthrough, but achievement by doing a real world, which is now called experimental image augmentation, non-virtual. So use 2 types of staining. HE and pan-cytokeratin staining. So HE staining, hematoxylin as eosin staining stains everything in different way, and pan-cytokeratin stains epithelial cells, which actually marks malignant cells. That's an approximation, but that's how it is, which is very nice because it shows you the growth patterns very nicely of malignant cells. And then we use color. We use the 8-bit, which is grayscale, and the binarized. So we used 5 thresholds for binarization, which is not like a pure virtual overproduction of images, but you see, if you the low threshold with only these are black and white images, so the black would be visible only for very strong staining. So we accentuate strong staining, while a higher threshold would actually turn black, also with less intense staining, which is kind of more obvious here with pan-cytokeratin, that these images look white because there's no strong staining, and then only here you have weak staining shown. So we have 14 images per from one patient, and this is not virtual, it's real. It's actually real. Yeah. So I will show you also how the code works. So this is done in raw code. So I'll try to demystify, because when you today you can find lots of information from my cousin GPT. I will tell you everything much nicer than me, but it's not good at pointing out what is important and what is not important, So the thing is that you have to choose, to work in code, you have to choose the platform which is most acceptable for a beginner, most compatible for a beginner, and I will show you such platform. And now I'll give you a brief overview of how the network actually learns, which is quite amazing and creative process. You can change parameters and indefinitely, and learning mechanism. So we have a pre-trained network, and we do a 2-stage fine-tuning by this. We just replaced it, this final layer, just erase it and put a new one, and then train it on our images with this newly corrected layer by a very high, relatively high learning rate of 0.01. And the other layers are frozen. That will be done for 50 epochs. And then the best epoch is recorded and further fine-tuned on all other layers after they are unfrozen. So the second stage trains both this final layer and all other layers, but the learning rate here is still 0.01, but the learning rate here is much lower. So why is it so low? Because this network is pre-trained. It had, with these 50 million images, it learned very valuable information. So we don't want to erase that information. We just want to add, if possible, a little bit about our specific images, but we don't want to erase its information, because that makes more damage than good. So then that's why we do this 2-stage process, and this is a usual approach to pre-trained networks, and a very low rate. But sometimes this actually second phase probably doesn't add very much, because the training of 50 million images was quite enough for this network to do a very good job, or actually the best job, without pre-training. So this is a classification layer, and its training is very useful. Its training is very beneficial always, and it cannot it cannot erase any of the pre-training valuable information. So we got another video here. So I will show you how It works in Google Colab because it's very beginner friendly. You actually just put your samples in, but first you have some code, and code has to be somewhere. And this code has to find your images. And it's so much easier if the code is in Google Colab, and Google Colab is directly connected to your Google Drive, and everyone's got to Google Drive, and that's I just put one folder called no meta and one page without metastasis and with metastasis. Those are outcomes, that already labels the outcomes, and this is the code. This is the three codes that I have, one for training, one for testing, and one for extracting, which I won't show today. Extracting the information about which should explain what structure actually contributed to the network's decision, but that is very controversial. I won't go into that detail today. So you just have, I think in this case, TIFF images, folder with TIFF images, one and the other TIFF images, and the the training step starts with opening the code. This is to do that all in Google. It imports the necessary software, and we direct the code to where our samples are, and our samples are in the data folder. We just say samples are in my drive data folder, and it will go there and find it. That's quite amazing. And even more, what's also quite essential is that Google provides you hardware. Yeah, you can get hardware from now from Kragujevec or whatever, but this is all integrated, so you don't have to do anything. You just choose what you want. 800 GPU is a very, very advanced 80 gigabyte graphics card, which speeds up like this type of deep learning a lot. It has to be GPU. It should be GPU, while TPUs are actually CPUs, which kind of emulate GPUs to some extent. Some of them are quite good, actually. So you can just try and see what works best for you, and also choose, depending on pricing, how urgent it is. So if it, if A100 does the test for 15 minutes, but if you can wait for an hour, or if you probably can wait for 12 hours overnight, but it would be much cheaper. So then, yeah, whatever. You just choose your own platform. And that's so this actually GPU platform, which is very nice, gives you 22 gigabytes of video RAM, which is about few thousand euros, and system RAM of 50 gigabytes. And here shows this space is 1.9 points per minute, which is 20 US cents. Which is quite cheap. It's quite cheap. It would do it in half an hour. Yeah, for some more complicated, it could take 10 hours, but still kind of it can work for $2 for in a very complicated setups. And yeah, let's see what it does. So it will first mount. It will go to the data. It will find the images. Import the necessary packages. This kind of exotic code-only software works with funny packages, which exist probably also in Microsoft Office, but they're behind the scene there in backend. You don't see them. But here you have to know all the weird names. Oh, sorry. I just... all the weird names like Torch Vision, Clear, and NumPy, and yeah, which you shouldn't actually, because that should be adjusted for every scientist. But for some reason, I said that I take it as my mission in life to actually produce a CNN, actually artificial neural network with graphical user interface, and I'll probably do it next year. It's not very hard. Oh, it always starts with me. No, this is the whole thing. So it's very actually nicely visualized that after it uploads images to the Google servers, and then it does one epoch after another. So this is a bit fast forwarding. It's not that fast, but it tells you for each epoch, the, let's say, validation accuracy is say 82%, and F1 is 89%, which is already quite high. So quite high. And this is training. Training is always, of course, very high. And then it gives you a loss number, so loss data, which is very yeah, this is the first stage, and then in the second stage is actually does the same thing. It's again 50 epochs, and I will stop here. It actually gives you the final result based on two stages: is 88% accuracy and 93% F1. F1 is a bit more strict than accuracy, so it's good that F1 is actually much is higher. So this is actually fantastic data. So to even completely separate training from testing, these two processes are even in different files. They're in different codes. So the test set, the testing is actually much faster. You don't need GPU anymore. And I will just go very quickly through it. And yeah, it just gives you the score, which is quite similar to the previous. It's 91% accuracy and 95, almost F1. It's slightly better. Yeah, so that is that was 8-bit. That's that we do. It was a single, was only with one type of images. Is actually the pancytokeratin stain. That's it here. Grayscale images. AUC, which you didn't see on the previous slide, and we actually calculated later, is 98, 0.98, which is very good. And so this was already very high. These individual versions of images provided quite good. Even the worst one was 0.91, but put together, they produced then when all of them were actually fed at the same time in the same network. So this was not ensembling like I showed you for this machine learning platform, where the result of each model was kind of averaged, and that was that produced the assembling result. No, the network was fed with everything, and it produced only one result, which was 100%. So that's quite amazing. That's what experimental augmentation does for you. It is complicated because you have to stay in the slides and you know, do a bit more work than usual. Yeah, thank you. That will be all. It's almost noon. So
thank you, Marko.
I hope that was useful. They think it was useful. Oh, obviously, yes, amazing. Yeah.
Okay, thank you for your exciting presentation. And does anybody have a question for Marko?
I have one. Thank you so much for this. Was a great overview for and the user-friendly. I think it's really important to have these kind of GUIs so that people can use them without coding skills. I have two questions. One is for this file that you've mentioned. Can you download the model and can you apply it on different, how closed it is? The final model for the classifier.
What do you mean, how closed?
How close the GUI is. So can you download the model essentially and apply it on a different platform?
Oh yeah, that's the link. You can just go to.
No. I wasn't thinking about the software. That's the final algorithm with all the features. So the model that it selects for classification. Can you get that? Get that one?
It doesn't select any models. It just produces all the models, and you select the models.
I know. Can you download the model that you want to select? That was my question.
No, it actually gives you it produces a massive folder with all the models. So if it makes
it's already there.
2,000 models, it will give you a folder with everything you can relates to that model, like absolutely everything, which makes it useful in my view. That's quite a lot of that's absolutely everything.
Great.
It gives you detailed results at the training, validation, test step, like just about everything.
And the last one, I mean, in Google Colab, you can probably specify anything you want, but it's using traditional kind of tools that were developed by the engineering community. And you nicely said that these were initially developed and trained on image recognition analysis. This is something that I kind of always struggle with, is that these algorithms that come from the engineering community, they are made using their terminology and they are focusing on precision and recall. These are not necessarily, and F1, these are not necessarily the parameters that
but
in the
that's
clinic.
because I collaborated with this code was made by professional, not only professional coders, but like a top AI lab in Canada, and but they don't know about ALK. They didn't know about ALK, and they knew about those, and then they did that, and then at some point I begged them and asked them to calculate ALK. So that's why I don't have a code with ALK. They send me the first code, but not the second code. And but you can introduce
all metrics that you want.
Google Colab is just you can put any code, whatever you want.
I just wanted you to comment on this so that that younger people can realize that we need in biomedical field different markers of performance, that usually our traditions are sensitivity, specificity, positive and negative predictive values, and that we need maybe to ask for collaborators to provide those metrics. And
Phi actually provides you with probably 20 different evaluators. But it's just like some that I never heard of. But then later with some networks, which I didn't show here, I also instructed to produce also loads of evaluators, and that works. It's not a problem. So I just run this original code. Actually, however, that code is a bit simpler than people would advise. It actually works much better. So if you make it more complicated, you can actually instruct the network to look for optimal learning rates for first stage and second stage. But then it works. It doesn't work as good. That's it works for days, but not as good. That's quite interesting. So here you manually actually select the learning rates, and it works much better. But there's I could play a lot with these. That's
Thanks.
a good actually exercise against boredom. And you can walk the streets, and you can monitor you can open your Google Colab notebook on your phone. So you don't ever have to stop.
So Miljana
Yes?
has a question.
Thank you. Yes, I think we've learned a lot today as well about this approach, and Marko has been doing it for quite a while. It's been very useful for the project as we've managed to publish one nice paper and another one is in progress about the radiomics approach for locally advanced rectal cancer. So Mladen and Marko actually analyzed a lot of MRI images and managed to produce a publicly available data set now. I think that's very nice to say because we contributed to science as well by these new techniques that are now available at IRS as well. And we've been discussing a lot now how to progress further on. So Marko, I'd like to hear a little bit of your insights on how we are planning to merge now all the knowledge that we have from the omics technologies and from radiomics, which is emerging as the imaging type of omics. So how do we integrate now this data into something that's clinically tangible, clinically usable, like with the translational significance for application for the patients? What do you think? Where should we go from here?
Yeah, we could load all molecular, clinical, pathological, and imaging-related data either into this machine learning platform or into an artificial neural network, and just get a result, which integrates them all, which is quite amazing. You can even ingest. All these platforms ingest proteomics data just as nicely as anything else. So it's just a matter of imagination what you want to put in.
And I think we will need to differentiate well the scientific question, right? For this specific project, we were trying to see if we can find some differences between the responders and non-responders, both clinical and histopathological. So probably this is how, at least how I see it. You pose a question and then you tell the algorithms what they need to do, right? To find the differences by integrating data both from different omics and to give us this is yes, this is no. This is like the plastic view of the researcher.
Yeah. From this hypothesis side, you need some outcome. You need to define an outcome because neither of these will work without an outcome. And it's best if outcome is binary, although it's not necessary that it could be continuous also, but it's easier if it's binary.
So this cohort was around 71 patients, if I remember well, for the radiomics that you and Mladen did.
Yeah, that's why we couldn't do this type of machine learning. We did something simpler because of the size. And then it was up to Mladen to increase the numbers. So usually above 120, you can already do a little bit more to put some better technology at work.
Yes,
and as I mentioned, about 220, you can have a test cohort also, and if you have external test cohort, then it's like much that gives value, extra value to a study.
Perfect. Yeah, I mean, we need to have this external validation and a prospective cohort, preferentially not from IRS. It would be good also to test it on other cohorts from the partner institution. So I think we should continue this work. I think Mladen is at the clinic now. He was a bit, he connected for a while, but I don't see him now. So it's something we need to work on. And if anybody from the team who is currently online would like to join us, of course, you're more than welcome. Marko is the expert at IRS for these types of analysis. So please pick his brain. Thanks, Marko.
Yes. I usually in papers, I throw mud at molecular methods and saying that they're so inferior, with analyzing morphology, it's so much better, and so, but then I wanted to do here the same, but then I realized, this can also work with molecular methods, and then most of you do molecular methods. So, we should leave it open to integrating molecular, because so far I only integrated mostly clinical pathological, although these clinical pathological parameters that I inserted, a lot of them are molecular. A lot of them are also molecular. So let's say receptor concentrations and this and that. So lymphocyte counts, eosinophil counts, things like that, So there's basically no limit here other than defining an output, which is survival or responding or non-responder to therapy or something like that, or metastasis, no metastasis.
Thank you, Marko. If anybody doesn't have any questions for our speakers, I would thank first to Miljana and Marko for this great webinar, and to all of you that participated. So I think that we can also continue this discussion after we finish the webinar, and because we are a little bit late now, see you maybe on some other future things. Now when we finish the project, we will try to continue this educational stuff. So thank you, everybody.
Thank you.
Thank you, Miljana and Marko, and everybody. See you. Bye-bye.
Bye.
Bye.
Bye-bye.
Where do